Hello, everyone. Welcome to Foresight's Biotech Group. Today, we're super excited to have Mark Woodward here with us. Mark has a background in bio and machine learning and was most recently a researcher on Google's Brain team. Before that, he was finishing a PhD at Stanford. This year, he founded Wake Bio, which is a startup that uses machine learning to reversibly preserve whole organisms. So we're really excited to finally have a cryo speaker on again. And thank you, Mark, for joining us. Please take it away.
Thank you. So I will jump right in. You all can see my screen. Is that right? All right.
Yes.
So this is Lydia and Timothy. Lydia and Timothy were conceived in 1992. They were then preserved for 30 years using the wonderful technology that Dr. Fahey, who is on the call, developed. As embryos, they were preserved, and then they were warmed, implanted, and birthed in 2022. So 30 years of preservation. I think it's a strong example of how low temperature can effectively halt biology and without damage to the organism. So we'll come back to this, but this is just a wonderful little glimmer of hope. Okay, so death is a problem. There are just tremendous number of people die every year. It's sort of 6 World War IIs going on every year, 20 Vietnams. Every day it's like 60 World Trade Centers. But I think for most of us, we've just sort of normalized that death that's all around us. And largely it happens at hospitals, areas that we're not aware of. The assumption, I think, for most of us is that these people have lived a full life. They're ready for their death. They might be in pain. They're at the end. They're ready to succumb that death. Unfortunately, research doesn't show that's the case. So there was a study that interviewed basically 2,000 hospice patients. And I think it was somewhere in Europe and then also in Canada. The result was that basically 83% of those people who asked, and these are people that are in the last weeks of their life, they likely have some terminal illness, they've been in pain for a while. Most of us would think they have every reason to just throw in the towel, but they respond, 83% of them, that they would like to keep living. So there is a need, even at that very end, among people who are dying. Okay, so what's the challenge for Lydia and Timothy? Why can't we just scale that up? And the problem is one of literally scale. So in terms of radius, so distance to a cooling or warming surface, the blastocyst phase that Lydia and Timothy were preserved at can be on the order of 100 microns. And we are about 10 centimeters in that longest or in that shortest distance. And that's 3 orders of magnitude. So we are, substantially larger than what was preserved. And so, okay, so why does this larger scale have a problem? So this is the sort of flow of that problem, leading from that thicker tissue down to more toxic effects of cryoprotectants. Each of these arrows kind of has a physics issue behind it, and we can go into those. I will go into some of those, but we can talk about all of them. Essentially, that thicker tissue results in a slower transfer of heat. So this is kind of intuitive. You want thicker walls, a thicker insulation in your walls that the heat will come through at a slower rate. We can talk about why that is. But yeah, so thicker tissue means there's slower heat transfer. That slower heat transfer means the temperature of the tissue decreases at a slower rate than it does in the thinner tissues. That means that you end up spending more time in a dangerous, and we'll talk about this, dangerous region of temperature in which ice can nucleate and grow. So you spend up more time in that region, so more ice will form. Ice is really the enemy here. And then, because you're spending more time in that region, more probability of ice, you end up needing a higher concentration of chemicals that avoid the ice, that prevent the ice from forming. And then at those higher concentrations, these chemicals are toxic to the tissue. So this is sort of the tension we need to increase that chemical concentration to avoid the ice, but it ends up being toxic to the tissue. Okay, we will talk a lot about ice growth. So I just thought I'd explain kind of a key figure here. This is from Brian Woke's, one of Brian Woke's papers. I think he got it from somewhere else, but I'm citing him on this. And basically, so this is temperature on the x-axis. And then, so as you're starting from the right and you're going to the left as you're decreasing the temperature, there are two things that need to happen for ice to really form and then for ice to cause damage. First, it sort of needs to form. It's not just as you cross 0 degrees, you don't just immediately, or cross the melting point here. This is melting point of this cryoprotectant. As soon as you cross that, ice just doesn't immediately form. It's sort of a stochastic event. You have to have water molecules that get together at a sufficient radius to then be able to support and grow. So the first thing, but there are different regions for the ice growth and ice nucleation. As you go below the melting point, the first region, so you are in a region where the liquid or the whatever you're trying to preserve is not very viscous yet. It still can flow around. So if there was an ice nucleation point, more water molecules could join on. That's this ice growth region. Then as you go cooler in temperature, the viscosity increases, so the water molecules can't move as much, but it's more thermodynamically favorable to form an ice nucleus, their first nucleus. So you have this nucleating region. So basically ice growth, ice nucleation, and this will come in later, but yes, so ice is an issue. Okay, so what are some approaches that people have taken to preserving tissue? So there are hypothermic approaches. So this is above a melting point. So as you decrease in temperature, the chemical reactions, the rate of reactions decreases exponentially. So you gain exponential time. And in the limit of 0 degrees Kelvin, you gain infinite time as you're decreasing the temperature, which is wonderful. So even if you're going from sort of body temperature down to closer to 0 degrees Celsius, you're gaining a lot of time. The biological activity is slowing down. The proteins, the enzymes don't work as well at that temperature. So just things happen at a lower rate. Oh yes, I'm letting people in. Oops, I need to be doing that.
Oh, don't worry about that.
You got it. Okay, great. Okay, so then there's some technologies that even cooling down above a melting point, so not actually these long-term preservations, you can still get longer-term hours worth of storage. Or with human patients, you can actually get an hour worth of surgery with them by stopping their heart, no blood flow, anything at lower temperatures, but still above the melting point or freezing point. This device here, this is also doing like perfusion. Greg is on the call, will know the terms for this stuff, but it's keeping the organ alive and actually ventilating the lungs here, but at a lower temperature to slow down the biology. So that's one technology. Another technology this is the MGH Harvard group. They focus a lot on this. So it's a high-subzero cooling, and there's sort of two technologies here. There's a supercooling, so you're trying to avoid any ice forming and just kind of be in this metastable state or of avoiding ice formation. And then there's partial freezing, where you allow ice to form and you're trying to control where it's forming. So maybe just in the vasculature. As it's forming, it's pushing out solutes. So the solutes in the rest of the solution are increasing. And then they end up vitrifying. Oh, we'll talk about vitrification. But they end up forming a non-ice solid in there, whatever. But these only kind of get you down to sort of the minus 10 degrees Celsius, and they'll gain you maybe a day of preservation, which could be very useful for like organ transplants. Like even gaining a day might really alleviate the supply chain of that. And then there's sort of the most promising, what I believe is the most promising approach. And this is a picture from Greg Fahey's paper, who's on the call, of vitrifying kidneys. And you can see, so vitrification is essentially you're cooling fast enough that the water molecules don't have time to fall into the crystal lattice that is ice and grow. So basically you're pulling out the energy and they're just slowing down, and eventually they stop where they are rather than forming that nice crystal ice formation. So they are transparent at that point. You can see on the right, it's a very nice, vitrified sample. And on the left, you see the ice everywhere. Okay, so vitrification, and this is what Lydia and Timothy, you can correct me if I'm wrong, but I believe that's it's vitrification that's used for the blastocyst, the human embryo preservation.
Correct.
All right, thank you. Okay, so now we'll get into some more technologies. So we're diving into vitrification a bit more here. So for a long time, and one could argue that it's still the case, warming is actually more of a concern than cooling. So, and ice formation during warming. So even if you get to this phase here and you haven't formed any ice, you're in a vitrified state, You need to warm at a faster rate than you needed to cool at. And I'll explain why that is by going back to this. Okay, so as you were cooling, you went through this, you're going from the right to the left, and you're going through this region of ice nucleation and growth. But that's actually okay during cooling because not nucleation, you're going through this region of ice growth. And that's okay because there aren't any nucleation sites yet, or there are very few. So the ice crystals aren't really growing, even though they could grow if they exist, they aren't growing. And then you get down and into the region where ice nucleation events happen. But now it's so viscous that the ice crystal, even though the nucleation sites have formed, they're not actually growing because everything's so viscous that the water molecules can't get there. So cooling is actually fine, you can reasonable rate. But now when you're warming, the conditions are reversed and much worse. You are first going through a region of ice nucleation where that can happen. And so you get all these nucleation sites. And now as you're warming up, you're still below the melting point, you're still freezing, you're still in a potentially frozen state, or in a state where ice could form. And you have all these nucleation sites, and now you're going through the region where those nucleation sites can really grow and ice can grow. So warming for this reason needs to be done much faster, like several orders of magnitude faster. So for a long time, that has been kind of the issue, that you could get into a vitrified state, but you need to warm fast.
Can I ask a clarifying question about that two-hump graph? So that's in the context of a vitrification agent of some kind?
Yes. And
do those two peaks not separate when there's no vitrification agent? Like for normal?
Yes, there's a strong, so again, Greg is more of the expert on this, but yes, so this is, in talking with Brian Woke recently about, this was selected to separate those in a CPA agent or CPA cocktail that is specifically designed such that, or happen to show that they are very well separated, but generally they are overlapping. You're right.
Right.
But but it is still the case that the peaks are that you're first going through this region of ice growth and then ice nucleation on cooling. So the issue is still there. It's just not as dramatic as this figure in many CPAs. Makes sense. Okay, so convective cooling, which is convective warming, we're talking about warming here, and in trying to bring that vitrification back up and trying to do it fast enough that ice won't form during the warming. So the ice baths really, the warm water baths are just insufficient as the tissue is thicker to create the warming rates, generate the warming rates that we need. So then two new technologies, sort of electromagnetic technologies, have come out. One is using just sort of alternating electric fields, and this is 21st century medicine. So you have kind of two plates, like capacitive plates, and then you're rapidly changing you're alternating the potential of both of those, and it's changing the electric field, and the water molecules are trying to align to that. And that's, in theory, hopefully uniform throughout the tissue, so you can rapidly, uniformly warm that tissue. That's a wonderful technology. So they have a recent paper showing in some animal, some organ, was forgotten, that they can rapidly warm that and then implant it. And I believe it's a rabbit, but they keep it alive. Yes, so that's rapid uniform warming with the dielectric warming, and then magnetic nanoparticles. So this is more alternating magnetic field rather than the electric field. And this is work at University of Minnesota, largely led by University of Minnesota. And what they'll do is they'll perfuse the vasculature with a magnetic nanoparticle, and then they'll alternate the magnetic field. And then when they want to, then they'll vitrify it. And then when they're wanting to warm it, they'll provide an alternating magnetic field, and this will rotate. But instead of rotating water molecules here, they're rotating these little nanoparticles, or kind of vibrating them and then rotating them. And they're probably eddy currents, but as I've talked with them recently, it's mostly the rotational aspect of these nanoparticles. So that's been working really well. And they did a rat, reimplanted it, rat kidney, implanted it back in, removed the other kidney. So the rat was surviving alone on this vitrified kidney, this previously vitrified kidney. And that survived, I think it was 30 days or so until they terminated it. So a lot of wonderful progress. But Yeah, so warming is largely, it's not solved, but we have technologies that are very promising and should scale up to human size, size for the warming. So we are but we are still stuck with the cooling problem now too. Still, even thicker tissue does still spend time in the cooling region where ice can form. So we do still need to be adding cryoprotective agents to that to avoid the ice formation, ice nucleation, and ice growth, and increase the probability or increase the potential of getting into this vitrified state that's an objective. So there's a number of groups that have been pursuing sort of development of cryoprotective agents. It's not on here, but the most commonly best known or most well-known and used cryoprotective agent is by Greg Fahey, and it's called M22. And he's spent his entire career developing that, and it is the best performing. These are just other groups that happen to be using some high throughput experimental methods to attempt to improve upon that. So Wake Bio, my company, we have kind of two key focuses, two keys to our approach. The first is that we're focusing on whole organisms. We believe that with our end goal of getting to humans, this will reduce the chances of dead ends because we'll be experimenting with and improving on something very similar to our end goal, which is that the whole organism is preserved. We are starting with zebrafish and then scaling up from there. And then the second key to our approach is that we're kind of using this rapid experimentation and AI. So we'll be conducting experiments, hopefully up to 1,000 a day on zebrafish. We'll be changing. So the variables that will change are sort of the cooling and warming profile of obviously the concentrations of all cryoprotectants, but then the loading and unloading profile of those cryoprotectants. So when is each component of the cryoprotectant added, how long is it spent there, and what is the curves of those? So we'll be optimizing all of that. Right. And now, how much time do we have? We're great. So now I'll just show some progress we've made on different things, and then we can have some questions and answers. So there were kind of three. The goal is to get to this rapid experimentation loop where we're selecting experiments, running those experiments, and then updating the machine learning model with the results of those experiments, and then using that model to propose the next experiments to run. So using that machine learning in the loop as optimization. That is the goal to get to. Two important things before that. One is the sort of breeding of the zebrafish, getting a production of enough organisms to experiment on, and then the equipment to conduct those experiments on. I've seen a bunch of this, but the rest of you haven't. So this is the progression of the breeding infrastructure. It started as one little tank with just a couple fish, and then it grew, and now it looks like this. It's actually advanced from this a bit. There are automated feeding racks and automated processing of the eggs. So they're little green egg traps down at the bottom here. That's where they breed. And this one rack will produce thousands of eggs a day, which is great. But so that there's automated collection of the eggs and pumping up here are each of our tanks of different aged zebrafish. So the eggs get pumped up into whatever the youngest tank is there. Oh, this was an algae bloom that happened at one point. Okay, so right, the egg collection, we talked about that. This is the egg collection increasing over time. So now which I could certainly scale by adding another tank or just more fish, but we're above the 1,000, which was a goal. Eggs produced a day. This is sort of the processing this is a vision system looking at, or actually just a recording of this setup here where the eggs are pumped through this, and then a camera is looking through a slit that has a tube pumping the eggs through, and then there's a black background, and this is all to highlight the eggs such that they can be processed with computer vision. Egg, so you kind of see them there. Cool. So another major push has been the developing an infrastructure that would allow for a parallel array of cooling, each with their own cooling profiles. So experiments happening at the same time, cooling down to cryogenic temperatures, warming back up at any rate, at a rate above what we would need to avoid devitrification or ice forming on the warming. So we've experimented with a number of these. This is one approach to that. Essentially, the cooling liquid is in here. In these, I'll describe here these plots. It's actually ice water, but this would be liquid nitrogen to get down to the low temperatures we need. This is the same little well, I'm calling it here, where the fish would be in and the experiment would be conducted. And it can be dropped down, it can be lowered into the liquid nitrogen for the cooling process. And then it has a heating, basically wire wrapped around it. What is it called? Nitinol or something like that, wire wrapped, but it's basically like the wire that's used in electric heaters, wrapped around it for the heating. In this case, we are small enough that we can create a heating. We don't believe that we will encounter the ice formation during warming. So we don't need to introduce those other electromagnetic techniques for warming quite yet. So we can just do it with resistive warming or sort of conductive warming.
What is the size when you say small enough? Like how old are the zebrafish? What's the size?
Yeah, so that's a great question. So they are under a millimeter that we'll be starting with, but they scale up to 20 millimeters. Yeah, I think the adults are 20 to 40 millimeters long. And one of the things we need for this AI optimized loop is that the AI needs to be getting signal of things working along the way. So starting at a younger phase, so the sub-millimeter zebrafish, and then going to its later phases of development where it grows, it fits very nicely with the approach that we're taking.
Great.
Okay, but controlling this, so there's heating, warming, and we don't want to just plunge into the liquid nitrogen. We want to actually control the temperature. There might be annealing phases. One phase, we want to get it down to a temperature that we can load the CPA at. That one's not that big of a problem. But then just before we go through, before you enter the vitrified state, it's also called a glassy state. There's a glass transition that happens. And at that transition, there can be a lot of expansion. The material can expand. And if you're heating or cooling, if you're not uniform in your heating or cooling, then it can expand at different rates. And there's stresses that build up in the material there, and you can get cracking. So typically, or what I've been seeing lately that people do is just above that glass transition temperature, you'll hold the temperature steady for a while, and then you'll slowly go through until the temperature has normalized and these stresses have reduced themselves across actually, no, not the stresses. The temperature has equilibrated, and then you can go through that glass transition. So yes, you want to control the temperature profile. So controlling this kind of when do you apply heat, when you drop into the cold bath to hold the temperature or follow the temperature you want is not an obvious control mechanism. So we've used machine learning for this. The first 9 images here were just so we recorded a bunch of data of this, just random controls and what the temperature responded to those random controls. The blue line is how the temperature actually responded to it. And then we learned a simulator of that, so to predict how it would respond. And the orange line is the predicted response. So starting from this temperature, given specific controls of heat and cooling, how will the temperature respond? And the blue line is our prediction of that. So it nicely follows what happened in the real world. Then we took that simulator and learned a controller. So this is a policy gradient reinforcement learning controller of this. So the green is the desired temperature. And then these red here or blue red here are the controls that the learned controller is outputting. And then the blue is the actual temperature. So it's trying to get to the green. So here it's saying heat, it's applying heat, and that's driving up the temperature. And then before it even gets to the desired temperature, it cuts off the heat. And then that gradually brings it into the desired temperature. So that's really nice. Cooling, there are plunges. some of the issues here where you don't want it to, like if you just fed this to a controller, it would be happy to say hot, cold, change those really rapidly, but that's a lot of up and down. It can use up your liquid nitrogen very quickly or just be jarring to the system. So getting it to smoothly transition and have very few transitions was a difficult part of that optimization. Then we took that controller that had been learned in the simulator and transferred that to the real world. And this down here is data from the real world. So here the desired temperature is this green line. And so it's turned on the heat to drive that temperature up. And then it turned it off. Here the desired temperature changed. It again turned on the heater to drive it up to that temperature. And then it's holding it at that temperature. So this learned controller is working well. There's a little pulse to kind of keep it at that high temperature because it was naturally coming down. And then there's a drop here, which you guys probably saw while I was talking. Okay, so we'll probably, I don't think that this is the setup. It won't scale to the number of experiments I want to have happening at the same time, but it's a wonderful first step, and all the AI or machine learning can be transferred to that, which is nice. Just wanted to show you this progression of just sort of what's it like to something as basic as trying to regularly feed the fish. So there are 3 different foods depending on their different phase of development. And they are, two of them are extremely sticky and they're like a fine powder, and they're hard to dispense in a regular way. The first, I just switched to another tab. Did you guys see that? Can you guys see where my cursor? You can. Okay, great. So this is
Yeah, we can see the feeder side.
The 3 feeders, or you're seeing 1 feeder?
3.
3. Okay. So I did this progression. I don't know. I think that this stuff is interesting, so I will share you as well. So this was the first attempt. It had like an auger style in here, and there's a motor on here, and it would push the food out. There were a number of problems with this. One was there's an air gap up to the food, so the food can absorb moisture, and you're wanting to keep it pretty dry until that. It's also really sticky. So it would stick on itself and then drop out in big clumps. So the portions that you were portioning out were not regular. So I had to move on from that one. Move on from that one to this guy. So this guy, I think, if you can see in here, there's a disc that rotates and it has a little hole cut out in it. And that controls the portion that you're dispensing really well, which is nice. And then there's a hopper up here where all the food goes, and that drops down into the hole. And then here there's a vibrator motor that like helps to encourage it down, which worked great in development. And then, so I created the system, put it all up, and then what I hadn't ever done is waited a full night and tested again. So it fed the first time great. And then the whole next morning, basically the food had like compressed down. And I actually think the vibrating might have helped that compression. And it formed a little bridge over and wouldn't dispense out anymore. So this design decided to move on from that one to this final one, which has been, well, I hope it's the final one, has been working really well. So it still has the disc for the controlled portions, but then inside here, it has a little paddle that kind of, so this moves around and it catches on the little, I don't know, tabs there that it'll catch on and pop. And that really breaks up the food and moves it around. So no longer is a vibrator needed, the vibration motor thing needed. And it all works well. And then there's, over here, there's a little, so it rotates out, and then there's a ball bearing here on a spring, so that kind of pushes it out. So it's like super robust and kind of works, but it's so funny. We're trying to do this cryobiology, but end up inventing this. I think probably drug packaging systems have solved problems like this, but yeah, we created our own. So that's that. Okay, so that's working really well right now. Yeah, so this is the second to last slide. We are experimenting with the kind of inductive warming approaches. This is not volumetric uniform warming, but it's experimenting with the electronics needed to create a system to inductively warm something. Up here, what you're seeing is an example of those thermal stresses. So that's actually not, what's formed here is not ice. It is stresses that have built up as it's cooling. This is propylene glycol in liquid nitrogen, and stresses are building up, and then they just build up to a point where they pop and they all release at the same time. And you certainly don't want that to happen in tissue. And that's caused by non-uniform cooling. Similarly, you would have it in warming, non-uniform warming. It's like dropping ice cube in hot coffee or something, it'll crack. And you don't want that. Yes. So yeah, that's another problem that just needs to be avoided. All this stuff is the machine shop. That's the liquid nitrogen supplier. This is my daughter, Steffi, looking through the microscope, and she set that up. It was cute. That's that. We are between rounds of funding, so that could be a good time to get in. The valuation doesn't yet reflect the progress that's been made. But yeah, so would love to have you on board if you're interested in investing. Or yeah, interested in anything, happy to talk. And we can take questions now or whatever you'd like, Lydia.
Yeah, let's start the Q&A. Thank you so much for that great presentation. I think there is a question from Steve in the chat.
Okay.
Already.
Yes, I'm trained in organic chemistry, and I know that there are bonds that can be stimulated by infrared light that affect things like rotational and vibrational frequencies that would turn into heating effects. And I was wondering if anybody had studied the frequencies of ice that are apparently different than the frequencies of water, so that the formation of ice crystals during the warming process might be directly mitigated just by something as simple as illumination.
Yeah, I suspect, so Greg can jump in and give the real answer to this. I suspect that depth of penetration would be the issue with wavelengths that are near, well, actually, I mean, you can see through, I mean, vitrified samples are transparent, but the tissue itself is not transparent. So I'd assume it's just the depth of penetration that would be an issue, but I actually don't know if people have researched that. I've not come across it.
Yeah, I think that your answer is correct, Mark. That's a very astute question, Steve. I found myself asking the same question when I was an undergraduate student at UC Irvine a long time ago. And in theory, yes, if you had a small enough system so that IR waves could heat the ice selectively, that might be a very interesting thing to do. But unfortunately, as Mark was saying, IR penetration depth is only a couple of millimeters, a few millimeters. And once Mark gets to bigger systems, he's going to find that the IR just doesn't go deep enough. So if you want to know more about that general sort of problem, you might want to look at one of the more recent issues of cryobiology. Mark mentioned that Brian Woke, W-O-W-K, published a paper recently going into great detail about how the penetration depth of electromagnetic energy depends on the wavelength, and how he's been able to select the right depth for large organs. And as Mark said, we use that system to vitrify a rabbit kidney and transplant it, and kept it alive for a couple of years after we transplanted it. But it was a very sharp question.
Yeah. And I just remembered too, the early zebrafish embryo preservation work at University of Minnesota, John Bishop's lab, they used laser warming. So similar, I mean, it's light and it's warming it. They then moved, I think it was damaging to any pigment in the embryo would absorb at a higher rate. So it was blinding them and doing other damage to them. So they ended up moving on to what they call a cryo mesh, this sort of a mesh technique, but they did experiment with the laser warming.
So Mark, I guess I have a question for you, and
Yes.
I can reveal myself here again briefly. Let's see, there we are. Yeah, so my question is, have you gotten to the point of screening some of the cryoprotective agents against your zebrafish and seeing how well they respond?
No, not yet. No, the latest point we're at now is, everyone could hear this too, is a design for a system, like an array of these heater cooler. I think the feature I really wanted to have, and this was talk with Laksha Ganwar, he's a young researcher. We really want to have to control the loading, and controlling the loading even on a component level. So if you wanted to, you had mentioned this in the past, but even you can do this if you even want to do a pre-treatment phase where you're stressing it or something, you could do that. But even once you're doing the loading, like it might be the case, you know better than I on this, but the different components it might be important to load them at different times or different rates. There might be some benefits to that. So creating a system that would be an array of these 50, it's kind of 49, sort of 7 by 7 or something of these experiments, and they're all independently controlled. Each can have their own schedule for each component of the CPA. So that's largely designed now, and it'll be and the next steps are de-risking every one of the components. So there's a little valve for spraying we'll switch to a spraying of the liquid nitrogen instead of a plunging of it. So they're de-risking that. Can we deposit at a rate needed and at the small enough volume needed for ramping up the loading and unloading of each of the CPAs, and even the thicker CPAs? That's a concern of like, can I dispense a small enough quantity of that to create this ramp? Yeah, because we won't be doing like you guys do, I believe, with the organs, where you're perfusing and you have a constant flow, and then you have these peristaltic pumps, and you can really control the rates of really the CPA itself versus whatever the carrier solution is. And so you can control that, but running all those lines to each of the experiments would be too much. So we're probably going to do it in a way where there's a dispenser, and so you're mixing and then you want to mix them before the organism is exposed to that. So there's like an experience, sort of a mixing phase of that too. But yeah, so it's a lot of things going, but all that's been designed and now it'll start to be there, but no experiments yet.
Okay, so in order to do that kind of high throughput screening of cryoprotectant treatment protocols, do you have it set up so that the output of each one of those protocols goes into a separate chamber so you can evaluate each outcome separately?
Yeah, so I'm really open to suggestions here. The outcome that we'll be measuring is just either heartbeat of the organism at the end, whether his heart starts beating again, or whether it starts swimming, so some sort of activity. And that's a very sort of gross measurement of it. And then the entire organism will be disposed along with a solution is at the end of that experiment. But no other analysis will be done on that, at least planned at the moment. But open for suggestions.
Mm-hmm.
Yeah.
I don't want to ask all of the questions, so I'll be quiet for a while, see if anyone else has a question.
You're muted, Martin. Don't look muted, but we can't hear you when you speak.
If you want to type it in the chat, that could also work. In the meantime, is there anyone else that has questions? Hit it, Gail.
Yeah, this is a business question, and that is that you're between rounds of financing. Where are you getting resonance in terms of investment?
So there are a lot of no's. I mean, 99 points say that I've had hundreds of conversations and there are 3 investors. So whatever. So it's high 90s of rejection. And I don't know what that is. I mean, this is only a sample of one. I have not done that many funding raises in previous companies. I suspect I'm being very honest about my end goals, that it is, cryonics, which I think a lot of people don't. Even people within the rejuvenation community, I think those communities, I feel like they should overlap, but maybe there's some reason psychologically why they don't. People don't want themselves to be healthy now versus don't want to actually die. But yeah, so I don't know. I don't think there's any pattern among those 3. 2 of them are just people that had experienced my work in the past and were anything you do, I'll invest in kind of people, which is wonderful. And then the other was interested in cryonics.
Okay, so have you contacted people? Are you able to contact people who are, say, within the Alcor community?
I have had, yes, and have a number of conversations.
Okay.
Yeah.
But they haven't been particularly magical in terms of, no.
Producing. One of them has like a standing, I don't know. Yeah. One of them who's on the board of Alcor will put in 100,000, but it's contingent. Actually, this is the way it goes when you fund, you're probably very familiar with this. A lot of people will say, I'll put in money as long as some top tier investor puts in money, because they're relying on that person to kind of do the due diligence. Yeah, that's right. So I have a lot of those, and one of them is on the board of Alcor that's like, if you get one of these, I'll put it in.
Carl has his hand up.
So does your search with AI imply, and maybe you said this explicitly, but I don't catch it. If so, imply that you have no confidence in Alcor and Tomorrow Bio's current preservation? Because
No, so,
we need to do better in the future?
I mean, I think, no, I wouldn't say that I have no confidence. I guess I would like to see, like, I will have confidence in a cryo solution when I'm seeing primates being preserved, reversibly preserved, coming back, then pigs, something that's the size of a human, coming back, seeming to be healthy on all tests. So it's that reversible aspect of it. I don't know that we've tried on an actual human to bring them back for that. So, I mean, it might be the case that M22 gets us the entire way and some other technique. It is certainly one of the, like, that composition will be early in the experiments that we do and trying to iterate from there. But yeah, I guess I just want to roll up to that point where I'm seeing reversible large mammals.
2 questions, Mark. One, do you have any intermediate steps before doing whole humans that you're thinking about from a commercialization perspective? And then the other one is, while you initially are going to be doing like very screeny, does the heart start beating again after kind of rewarming? Do you have any plans for actually checking for behavior or other neurological aspects in the zebrafish?
So the first plan, so no, I mean, the first plans are literally either basic swimming motion is that, and we can learn a model that's like captures sort of the distribution of what a healthy animal behaves like, and then getting a score of whether this is deviating from that. But first it'll be whether the heart beats, and then nothing is planned beyond that. What I suspect will happen, so, and you had a commercialization question, which I'll get to in a second, but so the AI will be able to optimize. It's unlikely that it'll be able to make large leaps. So even though it will understand the chemical structure of what's being fed in, there's still a real need for human scientists in the loop that are recognizing patterns that they've seen from recognizing something and a pattern that's in their head that will propose something that to the AI would seem very different. Pre-stressing, that's a good example of this, like increasing the heat before you start preserving to generate some proteins that help with the preservation process. That's an example that it's unlikely the AI would discover that or at least current AI as it's iterating. So scientists will need to be coming in, and then it's very likely that they will, for promising experiments that seem interesting to them, that they would take that cocktail and maybe even just take the organism at the end of the experiment and conduct some more detailed screenings of that. That's one answer. That's the answer to one of your questions. And then the other is, no, I am treating this with investors right now that this is and I think it's kind of I don't know, to me, it makes sense that it's a deep tech technology, meaning that it's something that is very difficult to do, might take an extremely long time, and most or all of the returns are really at that final stage. So this is the nuclear energy, what is it? Holy cow, I'm forgetting right now. Whatever. Fission, yeah, fusion, excuse me. Fusion power plants. Yeah, so it's of that level. I don't think it's of the same scale of funding that's needed, but it's of that same timeline and uncertainty and whatever. So at least the investors so far are under that assumption. I am still on the lookout for if there will be. I am cautious that will be a distraction from the long-term vision and that we would become a company that's then serving whatever that early revenue stream would be and not continuing to pursue the cryonics.
Thanks. I'm going to jump in here and ask Martin's question for him. Martin asks, whatever happened to the idea of using modified antifreeze proteins as cryoprotectants?
Yeah, I think that is, certainly something we're considering. I think Greg should answer that question. Anything we can get our hands on. We're not super interested in designing our own, or I would like to avoid synthetic chemistry. So identifying some molecule that we can't just buy off the shelf. Proteins, I think certainly peptides might be better for that. I'll give some of the answers that I'm really getting from Greg and Brian Woke. So Greg can jump in that the proteins generally, unless you're having them you're doing some genetic modification such that or some RNA transport where they're generating the proteins inside the cell, the size of the proteins means that they won't be getting into cells. So I don't know. So that limits potentially their use. I don't know. Greg, thoughts?
Yeah, there's a lot of things that we could say about this, but the main advantage of antifreeze proteins is to inhibit ice growth during rewarming after vitrification or during cooling, and also to promote supercooling if you're not interested in going all the way down the cryogenic temperatures. So if you want to inhibit supercooling, the best way to do that at the moment is to acquire the peptoids from Xiaoxi Wei's company and use those instead of real real antifreeze proteins or any other kind of artificial antifreeze protein that's known so far, because I think Xiaoxi's artificial peptides, non-peptide versions of antifreeze proteins are the best thing that there is right now. But that's only if you're interested in supercooling. Mark wants to go down the cryogenic temperature, so that's not really going to help him much. It might help him on the rewarming phase. One other thing that we might interject here is, as Mark said, these things are proteins, so they don't enter cells, so they can't inhibit ice growth inside cells. On the other hand, ice is not supposed to be inside cells. So if you either cool them slowly and freeze the solution on the outside of the cell, you don't have to worry about what's on the inside of the cell. Or if you vitrify, you don't have to worry about what's on the inside of the cell. So the antifreeze proteins actually don't have to penetrate in order to do what they would normally do. But by the same token, you also have to have other agents in the solution that do penetrate. And one of my biggest concerns about Mark's project is that he's chosen zebrafish, which live in the ocean, live in the water, which of course tend to be impermeable to water and other cryoprotectants. So he's kind of got himself into a little problem there, and he may be able to engineer some solutions, but it's a tough problem. One thing I will have to say about Mark, though, is he's obviously an excellent engineer, a fantastic machinist. He can design any kind of equipment and build it that he wants. So if he can come up with ways of handling zebrafish, he can be able to apply the same kinds of skills and expertise to another system that might be a little bit easier for him to preserve.
Awesome.
Yeah, and what attracts me about the fish, and Greg and I have talked about this, is the ease of production. So they fit well into the model. And as Greg has raised his concerns, we might use zebrafish as a demonstration of the ability to optimize some biological system, and not try to get to the full adult phase of that zebrafish, but just survive at a lower temperature than has ever been done before, and have the development of that survival done by this machine learning in the loop and the experimentation.
I think we have time for one more question. Micah, would you like to do the honors?
I feel bad making the last question for Greg, not Mark, but you said shouldn't be ice inside the cells. Why is that? There's water in there, right?
Yeah, okay. So as Mark has said, in order to form ice, you have to have two things happen. You have to have nucleation and you have to have growth. And he showed the example. And by the way, Mark, that actually was data that was generated by Brian Woke. So he did not get that from some other place.
Right,
It's actually M22 that you just presented. So in M22, the solution doesn't freeze because the nucleation temperature is way lower than the growth temperature. So you can cool to low temperatures. And if there is a nucleation event, nothing happens because the growth is impossible at the very low temperatures. If you look at what happens to a cell, the cell is thought not to contain any nucleating agents. So for whatever reason, ice always seems to form outside cells. It doesn't normally form inside cells. There were some really elegant experiments done on yeast cells. You can take yeast cells and suspend them in teeny tiny little droplets of water dispersed in oil, and then put those into a machine that can detect freezing events. And when you cool them down, the nucleation temperature of the solution, instead of being minus 40, which would be homogeneous nucleation for the water, might be minus 39 or something like that. So if there are any heterogeneous nucleating agents, anything inside the cell that would actually nucleate ice, it's incredibly weak. And under normal circumstances, you will always see ice formation on the outside of the cell first. Once that happens, then the medium becomes depleted in water. And so water flows outside of the cell to match water concentration inside and outside the cell. That depresses the freezing point of the cell. And so again, there's no supercooled water inside the cell, so the cell does not freeze. So I just finished putting the finishing touches on a review article that's going to be published in Cryobiology in the near future about the discovery of why cells survive freezing. And the reason they survive freezing is that they don't freeze. The medium around them freezes, but the cytoplasm actually undergoes a glass transition at low temperatures. So what's really happening when you freeze cells is you're actually vitrifying them, but you're vitrifying them between a big hunkin block of ice crystals on the outside. So it's a very interesting story, but that's the answer to that question.
Well, thank you so much, Mark. Thank you so much, Greg, for being here.
Yeah, thank you.
Really awesome to hear about what you're doing at the beginning of your project and your startup.
Yeah.
So thank you everyone else for your great questions and joining us. We've had a lot of biotech seminars in the past month, but I think we'll actually see you next month this time. And hopefully you all have a great rest of your day.